Yeah, welcome everybody to this deep meeting. Deep seminar. It's a pleasure today to have Meike Wortel from the Swammerdam Institute for Life Sciences at UvA, and she will tell us about her work on evolutionary coexistence in a fluctuating environment by specialization on resource level. Okay.
Well, thank you. It's a pleasure to be here. I'm Meike Wortel. I'm an assistant professor at SILS. And yeah, I'll tell a bit about this work of mine. And I know it's about emergence here. And in this case, this study is not so much about very many agents interacting. But although I have some ideas about that for future work at the end. But it's also about linking levels of complexity, because if you have an organism and you zoom in, there's a whole metabolic network, and you zoom in again, there's a smaller network underneath, and I think there's also some emergence in connecting these levels, but we'll see. So indeed, this for you, those of you who were last week, there was this discussion about the paradox of the plankton. So there's some theory in biology that only so many species should coexist as there are resources. And that's a little bit in contradiction with the fact that we find very many resources. Very many species on only what you might think are limited resources. So taking this further, or let's say to a smaller setting, even in a laboratory, you can grow species on only one resource. So that's been done here. So what you see on the left, that's an experiment from the 70s. And they had two E. coli strains. So that's a bacterium. And they were just slightly different. And they grew them, and it says transfers. So they would grow them until they ate all the resources, and then the next day they would take a small sample and transfer it to a new medium and grow it again. And what's plotted here is the fraction of the two strains. And what you see is that if they start with one at a low fraction or the other at a low fraction, so is one, they go to some kind of stable coexistence. So they only use one resource, glucose. So you would think maybe only one species can grow on this, or one strain. Strain is, let's say, a smaller unit than a species. And then they had some hypothesis why this might be. And the hypothesis was this. So they say maybe the species, they differ a bit. And here we have the resource and the rate of uptake, and one is just really good in taking up the resource almost at the whole range, and the other one gets better and better if there are plentiful resources. And they made a small mathematical model of this. So in this case, so in the beginning, when they've just transferred, there's a lot of resources. So then species 1 will grow better, but then after the resources are depleted, species 2 will grow better. So they made a small mathematical model and saw indeed it could be possible to sustain coexistence because of such a trade-off. And so why is this stable, this coexistence? That's because frequency dependent selection. So, well, we have these fluctuating environments. Well, this is just these two types. So we call the low substrate specialist, low S specialist, and the high S specialist. And those specializations might come from some underlying investments in different resources. For example, with many transporters, you might be good at low substrates, while with many ribosomes that copy the DNA, you can sustain a faster growth rate. And then, so every day these resources are replenished, but then what the profile looks over time, that depends on the species that's there. So the idea here is that the blue one, with the low substrate specialist, it takes a long time before all resources are consumed. While with the high substrate specialist, the red line, the resources are consumed quickly. So the environment that they see is actually decided by species that are there. And then the negative frequency dependent selection comes because every type does better on the profile set by the other species. So that's the red line with the blue border is going better than the red line. So the low substrate specialist goes better on the high substrate specialist resource profile. So in this way, you can have a stable coexistence, because if you get a little bit more of one type, it starts to grow less well, and the other one relatively increases again. I hope this is kind of clear, otherwise, yeah?
I'm only surprised by your word selection, because it's competition, right? We're talking about competition, not about an evolutionary process.
No, at the moment, no.
Okay.
Indeed, no. So it's just indeed, this is just an anecdote that's actually come soon. So indeed, so you get this dynamics all the time, right? Because every time you replenish the resource is eaten, these two types grow. So it always stays dynamic in a way. But we can still talk a little bit about a stable coexistence, because after a while, these dynamics stabilize. So the dynamics look the same every cycle again and again. So indeed, for example, in this, this is a long-term evolution experiment. So these are thousands of generations. Experiment. Also, they started just with one clone of E. coli, also only glucose there. And what they plotted here is all the alleles, so all the genotypes, so different types. And what I wanted to show here is that you see here, for example, for a very long time, so thousands and thousands of generations, you see this diversity still there. So this is also the same kind of setup as I told before. So of course, in natural environments, you also have fluctuations, like in the oceans, for example. But so then the question is, can this this specialization on low and high resource explain this diversity that's found? But then evolution comes in, because indeed we only looked here at the fact that the ecological system, so the two strains, they stabilize, but what if they can mutate and they can become different? So that's signified by the yellow strain there. So that's actually what I set out to do a bit, or is that, can you still get this coexistence of two strains if they can actually evolve? So can it be evolutionary stable? So, oh, I changed it to 4 parts. I haven't changed the title. So yeah, I start with something about adaptive dynamics that I've used to study this question. And then we need to know what the selection pressure is in such an fluctuating environment, what the evolutionary trade-offs are, so what kind of types can exist, and then well, finally we want to answer the question. So the idea of adaptive dynamics is to understand the evolutionary dynamics. And these are often shown in pairwise invisibility plots. So I'm using a little bit graphical method to get to my conclusions. So I'll explain them a little bit. So on the x-axis, you see some kind of trait value of the resident. And on the y-axis, you see the same trait value of the mutant. And then anytime, so do you assume the whole resident population has one trait? And then you look at all the different mutant traits, so that's like a vertical line, and see if they can invade, if they have positive selection coefficient. And if they can, you put a plus, or here they made it green. So you do that for every combination of resident and mutant trait values, and then you can kind of realize what happened. So here, oh, it's a touch screen, I guess. So here, higher trade values can invade. And here again, so you think that gradually, so it kind of has the assumption that trades evolve gradually. Gradually, you end up here at a high trade value. But if your pairwise invisibility plot looks like this, you have here what's called a singular strategy. And you go towards there. And then when you're there, you can look at the vertical line and no other type can invade. So it's called the evolutionary stable strategy. And now these can also be used to look at coexistence. There wasn't one isolate. So you have different type of singular strategies. So the one we saw was stable, but some are, this is for example stable but not attracting, so you'll never get there. And you also have the branching type. So you go towards this singular strategy, but once you're there, you can actually go both ways and you get diversification. So once you're here, actually all other types can invade. But then that's interesting, because if then different types invade, what happens then? And that you can use a kind of a trick, and it's called sweet evolution plots. So you can actually mirror this pairwise invisibility plot. So this is the mirror image. So then we have the mutant here and the resident here. And then you can overlay them with the original. And you get the area where you have both pluses. And that's the area where type A can, or type 1 can invade in type 2, and type 2 can invade in type 1. So it's called mutual coexistence, mutual invisibility. And that means you have some coexistence there, because they can both invade in each other's type. And so then you don't have a resident and a mutant anymore, but resident 1 and resident 2. And then you can also, with some ways, sometimes you can study the dynamics in those areas. We'll see a little bit about that. So I'm planning to use this to see if there's like this coexistence. So well, a little bit more on how you, so in all the books about adaptive dynamics that I found, they say here just use a fitness function with two residents, right? Because here you have two residents. You can look at the gradient of the fitness function and see in which direction it goes. But actually for this system, it's not possible to derive a fitness function for two residents. We'll see later. And you have to use graphical methods to figure out the dynamics in the coexistence area. So a little bit more on how this works. So here we have a pairwise invisibility plot with a resident and a mutant. So we flip it and we overlay it. So we get this coexistence area. So the area with two pluses. But now we want to know what are the dynamics in that area. And the trick is here that you use, so on the border of that area, that's also called the extinction border, there you have only, here you have only one resident, and here you have two residents. But it's all continuous over that line. So the behavior of the one strain, you can determine from the behavior just on the other side of the line, where there's only one resident. And if there's only one resident, you can, from the pairwise invisibility plot, determine the behavior. So to explain that, so here we have the red line, which is the Y extinction boundary. So type Y goes extinct here. So on the, so this is the coexistence area, and here you have only type X. And what happens for type X? Well, you can look. You can look just above the diagonal. Above the diagonal is plus, and below the diagonal is minus. So it means that there's selection for higher trait values. So that means that the selection goes this way. So that's what this arrow on top means. And here it's the other way around. So after singular strategy, it flips the selection. And the same you can do here. And then here, these small green arrows are just the sum of the arrows that you find around. So here it's down and to the right. So this arrow goes down and to the right. And then you can see that you end up at this singular point, which is on the line axis Y. So that means the two residents are the same. So that means here, to conclude here, there is a, let's say, an ecological coexistence area. So there's an area with mutual invisibility. So two strains would coexist. But if you let them evolve, they would evolve towards a point where there's only one resident type left. Okay, so one example of adaptive dynamics from a little bit of a different project. So it was about the chemostat environment. So that where you have like continuous inflow of nutrients. So yeah,
can you go back to that? Because I don't understand your last reason. Because at the x equals y point, so at the that's a branching, an evolutionary branching point.
No, it's not a branching point.
Is it not a branching point?
No.
Oh no, so no, then it's yeah, sure. Sorry, I was mistaken.
Yeah, we'll see an evolutionary branching point soon.
So that means that at the x equal y point, that means that there is coexistence, but they will evolve towards having the same trait value.
I wouldn't call that coexistence anymore.
They are the same.
Yes.
Yeah, so probably you lose one.
Yeah.
I would say that.
Okay.
So I would say there's no evolutionary coexistence in it. So there, I looked at yeast cells in the chemostat and how they will evolve. So, well, we have this setting where you have a continuous inflow of nutrients, a growth of cells, and then an outflow of both nutrients and cells, so a continuous flow system. And then we had a model of baker's yeast, and it's kind of a little bit simplified, but it has like metabolism. So it has glucose, a transporter. I don't know how good you are in the biochemistry, but that's then usually converted by glycolysis, which generates some energy. And then it can either be by respiration make more energy, or it can just be excreted to ethanol. And then also the cell with the ribosome, it can create more new cells at least, and it also needs energy. So we have like kinetic equations, a kinetic model here. And then I did some previous work where I showed that, so we wonder like what kind of type will evolve in a chemostat, and then you should grow as fast as possible. And in my previous work, I showed that in any metabolic network, the fastest growth is always achieved by an elementary flux mode. And that is a network that doesn't have a viable subnetwork. So what do I mean by viable? So we usually assume there's a steady state. So all the internal metabolites, they are in steady state. So they're produced at the same rate as they are consumed. And now, when there's no subnetwork that has a steady state, then it's called an elementary flux mode. So for this network, it's actually quite simple. There's two elementary flux modes, so the fermentation and the respiration mode. But the fermentation makes it ethanol, and ethanol actually inhibits fermentation, and that's extracellular. So when modeling, so that's why we discarded all the other options, and we only looked at those two network topologies. But we saw that when we have one of them there, the other strain can invade. And here you see if you only have the fermenter, the respirer can also invade. And it's supported by the fact that when you have a resident respire, actually other strategies, so these are mixture strategies, they all have a positive selection coefficient. So selection coefficient higher than 0 here means that they can invade. And at the time, we also made a small movie. So here you see the fraction of respiration versus fermentation. And here it's how much they invest in transport protein. And then we model both growth and evolution at the same time. And at some point, something will happen. So we start with a single type. And then you see after a while, you get one with that's fermenter. So it's a 0 respiration, but then if you evolve it for longer, then you see that there's also a pure strategy that has a respiration. You see a bit of a cloud around it, but that's because there are continuous mutations. But at the time we didn't use adaptive dynamics because I thought that the adaptive dynamics does assume that things evolve in small steps, and I wasn't so convinced about that assumption. Still, it does still work often when it's as bigger steps. So, well, I'll show it. Recently, I did the same analysis with adaptive dynamics here, but there was one other thing I wanted to show. So here we also have this negative frequency dependent selection, because the one strain makes this ethanol, this toxin that inhibits itself. So the more you get of that, the more you get of the toxin, and the more it gets inhibited. But there's also what's called a density dependent effect. So the more cells you have, the stronger this negative inhibition becomes. So you can tune the number of cells with the glucose speed. So you see, the more inhibition there is, the more coexistence there is. But then quick.
Yeah.
Oh yeah.
Can I ask you a technical question?
Yeah, sure.
You have this network, so you have a dynamical system, sort of called the E's or something like that, and how is the biomass growth? What is how do you model that?
Yeah, so there's actually 2 layers. So the chemostat is its set of equations, and then also this has all kinetic equations, but there's also enzyme investment. So the biomass growth is the biomass production over time. So the ribosome produces biomass divided by the biomass that's needed. So that's the investment in all the enzyme components summed up.
So, but because this is within cell, actually the network is within the cell, but you don't really model the cell cycle. Right?
No, indeed.
Okay.
You assume kind of balanced growth. So you assume everything is growing at the same rate, and then so you don't model individual cells. No. Just let's say have a function for the total biomass.
Okay.
I just want to show that I also did this then with this adaptive dynamics. So here I have a whole model and actually some optimization also in there, because you still have to decide for how much to invest in every of the enzyme modules. So I simulated this, but then we see indeed we have an evolutionary branching point here. So you go up to here, and then, well, it's kind of hard to see, that's why I made the zoom in, but you actually do split in one going up and one going down. So this trait is then the investment in these 2, in respiration and fermentation. And indeed, you can also make this or at least trait evolution plot, or at least mirror it over the diagonal to get this coexistence area. But that was a small detour, so we'll go back. So this was in the chemostat, but now I want to go back to the fluctuating environments, because in the chemostat we can easily say, we can kind of easily model growth. You don't have this temporal time scale. So fitness is just how much you invade if you grow faster than the others at that conditions. But in the fluctuating.
So the branching is actually possible because of the ethanol, right?
Yes.
Yes.
Yeah, because there's kind of a
But
toxic.
that's a second interaction there.
Yeah, there's two indeed. There's two. Here, there's two molecules. Let's say that. I'll come back to that at the end. So here we have this. So then quickly, so over in a fluctuating environment, we have to think a different how to define the selection. So first we model, we make a model of what happens over one such transfer. So we model growth, we focus, so you have normally you have a lag phase, then you'll have log phase growth. And well, at some point growth is inhibited because there's less nutrients. And then there's also stationary or death phase, but I focused only on the growth phases. And then growth is modeled with monokinetics. I don't know. So with substrate, increasing substrate, the growth rate increases. But it saturates, and it's sorry.
Double tap.
So it's characterized by two parameters, the mu max, so that's the maximum growth rate that you achieve at very high substrate, and the Ks, and that's the concentration for half maximal growth rate. So the lower the Ks, the better you do at low substrate concentrations. So this is then actually so I modeled that. This is the set of equations that determines that. So by this B bacterial mass over time is this monogrowth equation times the bacterial mass. And the substrate decreases with yield the same fraction, and then a yield which is a constant that determines how fast, how much substrate is converted into one bacterial mass.
You probably assume then that y is bigger than 1. Or smaller.
Well, I always have to think which yield, because you can define it both ways around, but
it's probably all the way around, smaller than 1.
Yeah, it also depends on what units you measure it in, of course, but usually you lose a lot more substrate for one bacterial mass. But yeah, of course, it depends on the units you take. So the y, that's also in the y component, to conversion of units. And then we assume the following. So we have these two specialists. So we have one resident there, and we assume that one mutant, one other type comes in. And then we want to calculate the selection coefficient from, we assume that there's only like one invader. So the whole substrate profile is determined by the resident. So we calculate the relative increase of the resident. Well, that's quite simple because that's only determined by the yield, because the resident just eats all the substrate. And then we calculate the relative increase of the mutant, which that depends on the whole substrate profile set by the resident. And then we check which one increases more relatively. So yeah, I have the results here. So the selection coefficient is the log of the bacterial mass of the invader. So the characteristic at infinity, time is infinity, divided by a time is 0. So that's the increase, and divided by the same for the resident. And then, well, the calculation led to this expression. So you see it's a function of the growth rate of the resident, so the mu max of the resident and the invader, and the Ks of the resident and the invader.
But wait, can because your equations are don't have a steady state.
No.
So Is this do you integrate this over the cycle of feeding
Yeah. So
Okay.
we integrated it from
0 to
infinity actually,
to infinity?
which case there's a lot of trouble with the reviewers. but actually at some point you can change you say change coordinates and integrate from the nutrient levels from the nutrient level you start at to 0, because that's the same thing, right? So from time 0 to time infinity, the bacteria grow and the nutrients go to 0. So you can also go from nutrients from there to 0. And then you can calculate that integral and then see how the invader growth does on that.
So what I was wondering, so you then, I mean, so I assume that what's in the brackets in the log, that's just the solution of the B star, and what's in the bottom, that's the solution of the B. But so you assume that the solution of B is not influenced by the presence of the invader.
Because you assume it's only like one cell of the invader, so that's kind of the effect that has on the nutrient profile is kind of negligible.
It's negligible at T is 0, but at T is, well, 24 hours or whatever, it might be.
It's still, I think the numbers you usually talk about, it's still kind of, because you start with like 10 to the 4 cells, if you then add 1 cell, still at T is 24, it's still a small portion. But maybe, so you have a point. Actually, recently a paper came out from some people who had a different solution to this, and they actually didn't take that assumption, but then, there's some you don't get a complete solution.
Of course, because then you would have to solve 3 component.
Yeah, and that's actually, I tried that for 2 residents, it's also not possible. So yeah, it's a little bit of the pity that this works only in this case. Also, if you say instead of time is infinity, I do it until time is 100 or something, it doesn't work anymore.
No, but I think that approximation is probably fine because all the scales are exponentially to 0. So yeah. Don't worry about that.
Yeah, but there is an issue that so depending on the profile of the resident, your fitness function might turn out to be positive for the invader simply because of the long tail and not because of what happens in the beginning. And so if you compare this with a kind of experiment where you have transfers every day or so, you might actually have the fitness of the invader might differ. What you predict on a long time.
You're right. I think we did do some. I did do some estimations of that, but I think for the setting it's now applied to, it's quite reasonable because in this experiment I started with, they transfer every 24 hours, and I think usually they stop growing already after halfway or less than halfway. And of course there might be some, but then you hardly notice any growth anymore. So I think the nutrients are depleted way before this 20. Of course, if it's exponentially, it never really depleted, but it's like as good as depleted. But it's definitely a point. And I did. Otherwise you have to resort to some simulations, which is, I think, okay.
You could simply do. You can solve it numerically, but not with simulations.
But does that help?
You can at least plot the entire pit, because what you're just looking at is a fixed point of a map from one point where you refresh to the next point where you transfer, and the map is only defined by an ODE system, so that you have to integrate numerically if you cannot do it out. But the map is defined.
Yeah.
And so numerically you can actually. It's pretty straightforward actually to draw that pip.
But then of course you lose the nice explicit solution. So
You look at the explicit,
Yeah.
yes, of course, but
But it might, yeah,
you can at least compare with what your whether or not the transfer interval.
I mean, it would be
Yeah.
a nice basic test indeed, just plot both curves, plot this curve, plot the numerically integrated curve, and
So
yeah.
we actually
Yeah.
looked at the competition in this batch culture of Daphnia,
Mhm.
and there is an influence of the transfer period. So you get
Yeah.
you get coexistence or not, depending on what the transfer period is.
Yeah, I can definitely. I did do some. Now I have to think a little bit to check it. I did do some.
But, we can talk about this later.
Yeah. I was wondering because I did check something. I'm just now wondering what exactly I checked. Because there were, of course, there are lots of assumptions. So maybe, yeah. Oh, this doesn't. You don't see it either. No, okay. Well, let's discuss it later. But yeah, indeed, you. Well, but just if, yeah, okay. I think that's reasonable. But for now, I'm going on with this simplification. But I have to. Because maybe it still helps with this simplification. At least we can go further, because this still does. This only gives us the selection coefficient, in which case indeed we can draw the pip. But first, we have to define what trait is actually on the x-axis, because we didn't really discuss that yet. If we want to draw such a pair-evasibility plot. So there I looked a bit at trade-offs, because if you can just go really well at low substrate and at high substrate, then surely you can just do better as well. So how do we know what trade-offs exist? Well, there are two ways I thought. Well, you can look at experimental data and just measure how well they go at low, how well they go at a high resource. Well, the nice thing is that it makes biological sense. Well, the bad thing is that it's not always easy to measure, the conditions are not ideal, and also we want to look at what species are able to do. So you want to look at species that are somehow a bit optimized, but usually, of course, you put them in an environment, they're not adapted to that. So this, my ideal case, you would be adapted to some resource concentration, and then you would go to another resource and you would follow the evolutionary experiment. But in practice, this is often what happens. You're somewhere here and you go towards the trade-off curve, but then you still don't have the whole trade-off curve. So then I decided on another way, and that's by using computational models of cells. Of course, these computational models have additional assumptions in themselves. But I thought they might still give us some insight. So this was quickly the process. So I go to a detailed model like the one I showed before, but also one that shows actually every reaction. So a more detailed model. And then I optimize those at a certain resource concentration. So what does that mean? Well, I set, for example, the glucose concentration to a certain level, and I optimize how much you invest in every reaction to get the best growth rate. And then I fix those investments. So I see those investments as an evolutionary parameter. Then once I fix those investments, I simulate it again over a whole range of substrate concentrations. So that's here. So for example, the blue one has been optimized as low substrate, the purple one at intermediate substrate, and the red one as high substrate. And then I fit a monocurve to that, and I plot all those pairs of parameters of the monocurve, the mu max and the Ks. And then I fit a evolutionary trade-off curve over that. So I did that. So this is the detailed E. coli model, more or less. So for those of you, here's glycolysis and the citric acid cycle. And these are then against all these elementary flux modes, of which the outer boundary is kind of the optimal possible optimal space. But then, so I did, in this case, I didn't optimize it. I just took all the EFMs because, yeah, that was a bit easier. And it shows kind of all possibilities. So this, I simulated all elementary flux modes over a range of glucose levels, plotted mu max, and this is 1 over Ks. So let's say you could see it a bit like going up, you're better at high substrate. Going to the right, you get better at low substrate. And fitted the curve. And I did the same for this model that we already saw previously. There I did optimize it at the range of substrate concentrations. Simulated it at every optimized model and plotted it. So now I obtained a set of trade-off curves. And then I just assumed now that you're always somewhere on the trade-off curve, so that you're somehow, let's say, Pareto optimal, because in that way I have a one-dimensional trade parameter again. And that also some yield also depends a bit on the metabolism, but I won't go into detail. So now I have this one-dimensional parameter. I have my selection coefficient, and I can draw the pairwise invisibility plot. So it looks a bit like this. Yeah, so you see that, well, this is, I plotted the saturation constant. So the higher it is, the more you're adapted to high substrate. It's always a bit confusing because the higher Ks, the lower your affinity. And because of the trade-off curve, the higher your mu max. And you can see that there's the singular strategy, and that it's attracting, right? If you're lower, so the black area means the plus, the invasion. So you go towards it, and from higher you go down. You can, I've also plotted here like the selection coefficient in that point. But then you can again mirror this over the diagonal to see if there's some coexistence area. So this is the mirror, and you see there's a very small coexistence area here. But we can also directly see that this exactly looks qualitatively like the example I showed before. So here we actually lose coexistence in evolutionary setting. So then I'm like, well, this was for one trade-off curve. And so there's also partly the conditions that you set. So maybe going quickly back to the equation. So what you can set, what you can set as environmental parameters are the S0, so the substrate at time 0, and also the B0, so the bacterial mass at time 0 here. So you can, those are environmental parameters. So I tried this again for different settings. So for different trade-off curves, a higher initial substrate, but actually everything looks qualitatively exactly the same. So that was a bit like, oh, okay, it looks like indeed if you make a toy model, you can find parameters where you sustain coexistence, but it doesn't seem so evolutionary feasible that you get this. But then actually, so that was the sub-conclusion. I did one point, so this was another experiment from the 90s where they again have the frequency over a set of transfers. And here they also looked into if this was this specialization on the resource level could explain this. And then they measured the parameters of the strains and they found, well, actually it cannot be explained from specialization on research level. And then they delved further into their strains and they found there was also cross-feeding or something. I think it was cross-feeding there. So one made something that the other one consumed. So it could still be that this specialization is important. It just doesn't sustain coexistence just by itself. But then I came to wonder at another question, because we are now talking, I looked at the trade-off curve of one species, but what if two species coexist? Can they then grow together because of this specialization? And then I thought, well, the problem is that they have both a different trait and how then to use the same analysis. But then I realized, so this is yeast and E. coli, bacteria. And then I thought like, well, I plotted those two trade-off curves, but here they have like the same affinity, but one has a higher mu max. Well, then of course that one will always, let's say, beat the same strategy of the other organism with a lower mu max, because these parameters define their behavior. So I don't actually have to look at this part of that species. So I only look at this part of species 1 of yeast and of that part of E. coli. And then I can plot it as a one-dimensional trait again. So I discard part of each species. And so here I plotted the pairwise invasibility plot, where on the left side of this line you are E. coli, and on the right side you have Saccharomyces, and here as well. So if we look at this part only, we look at E. coli versus E. coli, so it's the thing we saw before, right? And here we look at Saccharomyces versus Saccharomyces, so it's also a bit like we saw before. But then we have these areas where we look at E. coli versus Saccharomyces in these two areas. And then I did the same again. I mirrored over the diagonal. And then you get what's the ecological coexistence area. So here it's again the thin lines that we saw before. But in the two species cases, we actually have a larger area. But now we need to know what's the behavior in that area. And we don't have a fitness function anymore because we have two residents. So we go again to this graphical analysis, and I didn't find it. In the end, I found it in some appendix of some paper in 99, because there no one writes how to do that, because they think you just take the fitness function with two residents. So in qualitatively, the pairwise invasibility plot looks like this. So the one I just showed, you mirror it and you overlay it, so you get this type of coexistence areas. And then, well, we do here the same thing, but so it gets a little bit more complicated. So the singular strategies, they are kind of inherited, as you see here, because we saw like here, above the diagonal can invade. So this is the y extinction barrier boundary. So in the x direction we go up. So in the x direction we go up, and here in the x direction we go down, up, down.
Yeah.
But then there's an additional thing, and that is if there's here a vertical tangent to the extinction boundary, because at the extinction boundary you don't have x anymore. So actually, on the left of this extinction boundary, we actually have the coexistence area, if you look carefully. So on the right, we only have type y. So every selection coefficient for type X has to be perpendicular on the extinction boundary, because there can never be selection towards extinction. And that means that when there's a vertical tangent, the derivative in the x direction has to flip. Maybe it's a little bit, but anyway, in that way we can figure out all these arrows.
The red curve is the invasion curve of?
No, it's the extinction boundary. the Y extinction boundary. So this is the,
Okay,
so this is on the, so it's like here. So here, these types can invade. But if you look here at the mutual invasibility plot, it's the boundary between the area where only
where they both can go sit and
yeah.
well,
Yeah. So if we take all those things, we can connect the isoclines, and although we don't know the shape of the isoclines, we can see that some of the isoclines they have to cross. So if you have an isocline in the x direction and an isocline in the y direction, we have to have this point where there's no selection for either of the residents. So there has to be some coexistence in that area. And then I did some simulations. First, I started with only E. coli and only Saccharomyces, so I had some mutations. And on the bottom, you see the trait value, how it evolves. And there's continuous mutation, so you see that it goes to a fixed point. And also with only yeast, well, of course, there's always mutation, so there's always kind of a population. But then if I...
One short question. Are you here really doing evolutionary dynamics? So time is, you're always in stationary state, or are you actually solving the ODEs continuously?
A good question. What did I do? No, no, I don't think I have been. What did I do?
This top picture show these nice curves going up and down.
Yeah.
Or
This is just the alleles.
Oh, that's just the alleles. Okay, yeah.
Yeah. So just new colors, like a new type invading. No, I think I used what did I do? Because I have two residents. I think I did some simulation with the ODE. I think I did a little bit of a shortcut. I simulated the ODEs for a little bit and then extrapolated that. I have to check what exactly I did.
but you must be replenishing the resource here, right?
Yeah, so you have this
You have transfer.
yeah, there's continuous transfer, but these time scales are very long.
Oh, okay, so
Yeah. You have this continuous transfers. I just don't know. I guess I did some simulation, and then probably I simulated it over one cycle and then counted that for 10 cycles or so. Otherwise, it takes a long time. I know.
How did you introduce mutations? You just add some random perturbation to the mutant is dependent.
Yeah, I did add some at some point. Yeah, I used some code. It's not the most, I didn't try to make these simulations as good as possible, so it's maybe not the most solid way, but indeed there was always a chance to mutate, and then if you mutated, you have some probability distribution for the effect size of your mutation. Mm-hmm. And then there would be new types, and if you were lower than something, you could go extinct. But there was a maximum number of types. So if not enough went extinct, there was no space for, so it was like 1,000 types or something, maybe more, so there were a fixed number of types, but then there would be spaces, and there would be new mutants from, I think, the fittest type with some effect size. But the good thing was that I didn't really need the simulations here, because I already analytically show most things, so it's more for illustration. So I also started with both types here, and then you see they go to a stable coexistence. And then I wanted to plot this onto the previous one. So what I did is I kind of clustered, because you get this cloud, right? Because of the mutation, so I clustered them, and then I calculated the weighted average trait of each cluster to get, like, let's say, trait values for the different groups. And that I plotted here, so the trajectories go from yellow to purple, And you see this one goes to this equilibrium, that one to that equilibrium, and this one goes to some kind of state in the middle. So is it clear what I plotted here?
So the left bottom part is the E. coli picture we saw 2 slides ago, and then the right bottom is also the
right top with this.
Yes, the other, you can have got it in the S from 2 slides ago, and then left upper part, that part is then of 1 slide ago.
Yeah, indeed. And here I have 2 clusters, so I get here I get only 1 coordinate, and I plotted it, let's say, twice. So I always stay on the diagonal because there was only 1 cluster. And here there were 2 clusters, so that gives me 2 trait values, and so I have like an XY coordinate. And I go there, and here numerically I checked the selection coefficient of all possible types. And you see that only for the 2 resident types it's 0, and for all the other types it's negative. So just to confirm that this is indeed an evolutionary stable coalition.
It doesn't mean that the point in between there is an unstable ESS, or
which one? Here?
Yeah, that one.
Yeah, it is an unstable one indeed, but it's also a bit of a weird one because it's kind of on the boundary between the species. So there you cannot really mutate from one species in the other. So that was also kind of a complaint, but since it's an evolutionary minimum actually, or it's a minimum so you never really go there because it's just, you don't evolve towards that. That's right, it worked. And to at least support why it's kind of nice that we have an analytical solution is that I now looked at the whole range of parameters space, so the initial substrate and the initial biomass. And then I saw that if I vary that, so this was let's say the qualitative outcome first. I saw that if I vary that, then or it qualitatively looks the same, or it breaks up into either this one or that one. So it either this connects to this, or this connects to this. These negative areas connect. And then I did the same analysis for those cases and found there's no evolutionary stable coexistence. So then I can just change the parameter and see the qualitative looks of the pairwise invisibility plot, and from there determine if there's coexistence. So then I plotted the coexistence region. So I don't know if you could still do that if you have to simulate the pips, or that it gets very slow computationally, but maybe it's still possible. But I guess might be. Okay, so the conclusion here was that research specialization likely plays a role in species coexistence, or could play a role. Also when they are allowed to evolve. So then I was wondering, next a little thoughts about, well, of course, this was a very isolated mechanism here. You could also have trade-offs between the different growth phases and the lag phase, or the use of different substrates. And some other work I'm working on is that you have a trade-off between growth on substrate and resistance to a toxin. So in antibiotics, for example, you often see that one type has a higher growth rate, but it's more sensitive to the antibiotic. So kind of, the growth rate decreases quicker with increasing antibiotic concentration. So here on the x-axis, you would have increasing antibiotic concentration, and here increasing growth rate, while you have other types that grow slower, but that are much more resistant. So you could have and it would be kind of, I thought, interesting to see if different processes together can sustain diversity, and if you can quantify how much every process is contributing to this. So then I was looking a bit at a very recent paper from a few weeks ago, I think, on bioRxiv, where they looked a bit to this. So often these evolutionary simulations are done on what's called fitness landscapes. So that means you have a genotype landscape, so you can mutate. Your genotype can mutate, gives you some kind of phenotype and some kind of fitness. And there are all kinds of statistical models. You can simulate evolution, but then often there's no influence of the environment. But here for example, in the case we've seen here, that you influence the environment by eating a resource. So here, a very recent paper, they did include somehow this environment. So the idea is you have a genotype of zeros and ones of length L. So you have L loci, and then you go to a phenotype with two matrices, A and B. And one that gives the additive effects. So A just gives every loci has a fixed effect, and you just add them up. And then B is what they say epistatic effects. So epistasis is if one loci influences the effect of another loci. So there could be components there that look at the whole genome, and then that gives you some kind of phenotype. And the phenotype is then the vector of length R, which is the number of resources. And what's then your fitness? So your fitness is your phenotype on a certain resource, let's say, divided by the phenotypes of all the other types on that resource times how much of the resource there is. So this is kind of the ecological part of the model, is that how well you do on a resource depends on what all the other types are doing, if they are also eating that resource, let's say. And but they did some simulations in the, let's say, ecological and non-ecological setting. So in the non-ecological setting, this is just fixed, the fitness, and it doesn't depend on the other types. And what you see is in the non-ecological settings, well, so this is the relative fitness, and the how bright the dot is says how much the density of that type. And you see kind of a new type coming in and taking over the population. And you have 6 with one type. And then here we see the haplotype diversity. So of the non-ecological model, when there is like a new type coming in, there's suddenly a high diversity, but in the end, the diversity is almost 0. And the phenotypic distance is, let's say, how different the different types are that in there. It's also really low. But then for the ecological model, this is with 2 resources, by the way. For the ecological model, we see a very different picture with many more types. And here we also see the orange line. So we see that the haplotype diversity, which is it's around 0.55, I would think. And the phenotypic distance is also much larger. So there's actually more difference between the types.
So what does it mean for diversity to be a number between 0 and 1?
So the haplotype diversity, so it is the fraction of every type squared, the sum of the fraction of all the types squared. So you square every fraction and then you add it up. So I also had to think, because I thought there's 2 resources here. So I think about 2 species, let's say. I was wondering, then you have 2 times 0.5. So you have the 0.5 squared plus 0.5 squared. So you would get to 0.5. So this, because this is slightly higher. But yeah, it could be, of course, they say we don't really have a steady state because you have all the time these mutations.
So it means one is not diverse and two equal species are. So
two equal species are 0.5, but if you have 0.9 and 0.1, for example, what would that?
Then you're higher than a half. Yeah.
No, you would get lower, right? If you have 0.9 and 0.1.
Which doesn't exist here.
Oh no, sorry, 1 minus this you do. Sorry, 1 minus this. So if you have just one type of fraction 1, you get 1 squared is 1 and 1 minus that, so you get 0.
So that means that in this case, higher is more diverse.
Yeah. And we're used to the Shannon diversity measure, which I think is kind of similar. So I'm a little bit surprised by this definition, but maybe it's all a little bit what your background is. So these authors are really from evolutionary biology background. So maybe they came up with their own measure, which also works fine, I guess. But I would think so. I didn't see also the supplementary info was not there yet because it's just a preprint. So I'm just still wondering if you only have 2 types here, actually, let's say, I'm guessing that you have 2 clusters, like what I did, like if they would do the same clustering that I did before, and you also have kind of 2 phenotypes here, right? Because this phenotype vector has the length of the number of resources. You could actually plot it 2-dimensionally.
What I was.
Yeah.
So what does the word trade-off in the title here means? That means that the sum of the resource updates should be normalized.
Yeah, so they didn't do that, but what they did was, so you have these matrices A and B, and they did have a parameter that says the correlation between the resources. So they simulated it for a negative correlation, no correlation, and positive correlation. And if you have a negative correlation, you kind of get a trade-off, because then, yeah. So how this genotype affects then the phenotype, there you can build in some kind of negative correlation, and then you get a trade-off. But there's no hard limit, and they simulated for different. And they also found, so this result was for a negative correlation indeed.
If it's a positive trade-off, then you can get sort of Darwinian monster kind of idea.
Yeah.
Better, bigger the better.
Indeed, and they also said that, I think they said if there's a positive correlation, you don't get this diversity. There's more faults in it, which makes sense, right?
Mm-hmm.
I was thinking a little bit like, there is a chat. I see. Should I see?
Yeah, it was just
Somebody saying that's all.
Okay. So I thought like maybe it would be interesting to extend this with mostly this, maybe also this with like a bit more mechanistic model, in the same way that, so in the previous story, I've used these underlying models, let's say the underlying biochemical models, to determine the trade-offs. And of course you have to do some simplification with the model to get a nice function, but I think then you do guarantee that if you would integrate those models, you would get similar results. If you would, like for every step, optimize the whole metabolic model, etc. Still, of course, you would have to test that maybe, but of course you cannot have detail in all levels of organization. But so these kind of functions, I think they're mostly a bit made out of mathematical conveniency. And I wonder if it would be possible to replace them by an underlying mechanistic model, not meaning that to put in the whole big model, but maybe the results from like studies like the one I showed, and also with the antibiotic resistance, that you can see like the general behavior and then somehow include that general behavior in this setting. And also, this is, I think, often these kind of studies I don't. Well, research competition is, of course, a simple one, but you could also think that there are metabolites which you could interact with in a positive way or in a negative way, and then they will be toxins, and that you can produce them, which would be toxins for others, but also things that are toxic for yourself, or also you can produce things that have a positive influence with others, so you can also get cross-feeding. I was thinking, yeah, so I didn't do so much in this slide yet, but I think maybe it would be possible to include in a still a quite general way also other interactions and some saturating functions as well, because here I wonder, of course, maybe it's a good approximation, this, that you just divide it by everyone else who's doing the same thing, but I'm wondering a bit, if you're just better at it, does it matter that everyone else is also doing something like you do? Because they say it kind of resembles a chemostat environment, but yeah.
And everything here is also again, and everything is stationary here again.
Yeah. So the fluctuations is another story. Yeah.
Yeah, but the fitness they may depend on the inflow.
Yeah, which they actually kept constant throughout everything.
Which I find.
Well, inflow, actually, I say inflow. They say amount of resources.
So do they explicitly simulate resource dynamics?
No. This is the only ecological thing in there. The fact that your fitness depends not only on your phenotype, but also on the other phenotypes in the environment.
Oh, okay.
But the specific concentration at a certain point in time that the new movement is in, that's not.
No, that makes.
If they assume it's always the same amount.
No, yeah, so that's why I said inflow. I see it a little bit as inflow, because it's your fitness depends on how good you are on a certain resource, what others are doing with the resource, and some amount of resource. So I see it, if you think about the chemostat, I see it a bit as inflow. They actually don't call it inflow. It's or inflow into the system, let's say. Because I could think like if certain amount flows in and then you have to divide it with all of you.
Yeah, next one. Yeah.
Then this is like your part of the cake, let's say.
Mm-hmm.
And then S is how big the cake is, let's say. Yeah, this was actually the end that I had no idea about. Oh, sorry, I closed the presentation. That was not my intention. Oh, sorry. I don't know if there's any questions. I can open it again.
Okay, that's lots of questions, but we can have more questions for the group.
I was wondering about one of these pictures. So at some point, you have this double invasion diagram where you show the dynamics along the two diagonals and then the.
How is coming? Oh, double invasion.
This one. Yeah. So a little bit. One slide. Yeah.
This?
No, in the other direction. You just one of the last slide on the. One more. Yeah. No, the other direction. Sorry, this is not.
This.
Oh. No, one more. Yeah, this one.
Yeah. Okay. Sorry.
So this was sort of fixed point where it's going to the left up corner. Yeah, left up corner is. You said it's sort of at the crossing of, you call them isoclines. I would maybe no clines or whatever. It's probably mean the same thing.
Yeah, no clines maybe better.
Yeah, but
it must be at the crossing, right?
Yeah. But are these things? Can't you just find them numerically? I mean, probably computing by hand might be, but this is a zero of some.
Yeah, I've tried a little bit, but maybe I'm lacking the knowledge for this. So but indeed, finding the ways I try to find them were not faster than just doing a simulation, kind of. Although, because actually what I wondered was, because I felt like it's almost on the crossing of this one and that one. It's quite close to it, right? It's not there, maybe in earlier pictures I thought it was. And I thought that had something to do with it, but
Oh, you mean that these outer lines or no lines are just vertical and horizontal lines?
Yeah, but it's not true. It's not exactly there. And then I asked someone, and he said, who started this adaptive dynamics a bit, and he said they're often close, but there's no reason that they should be there. I don't know. Yeah, of course you can search the whole area, or you can follow some kind of way. You can do some optimization over that area and find the points, but it has to be some numerical method, I think.
There's a similar way to get there in the bottom right area.
Oh yeah, that's exactly the same. It is exactly the same.
Oh, it's the same because it's mirrored.
Yeah.
Yeah.
As soon as you mirror them, everything is the same there as there. Yeah.
Mhm.
Yeah.
Oh yeah. And this type of points is it very difficult to verify experimentally, or do you have some
it's
setup where you can show this?
It's quite tricky actually, because we're talking about evolutionary optimum. So in theory you would have to do an evolutionary experiment, which already comes with trouble, especially with more species. And also, yeah, of course, this is assuming we have a correct assessment of their evolutionary trade-off, but yeah, I think you might, and that the only way they interact is through this resource. So it's definitely some simplified assumption. But I do, yeah. So the way I see it, maybe more as an idea of what you could expect. And also, if I would do an experiment, I would first study, take a bit more complicated, let's say, realistic models with less assumptions, and then try the simulations. But you could think it can be useful to see like what range of parameters would be useful to check, for example.
Because I'm not sure whether I understood 100% completely, because later you mentioned something that if some parameters are changing, then this region becomes different or disappears.
Yeah, so this is for one set of parameters, and the parameters I mean is like how much nutrients you add and how much bacteria you add. So I assume that always the nutrients and the bacteria at the beginning of a cycle are constant.
Yeah.
So they stay constant all the time. So I did vary those two parameters. That you are able to control very well. What you are able to control less is that they don't interact in another way, for example, producing ethanol, etc. And that they actually, so the evolutionary trade-offs I found, I didn't go into much detail, but some of them are actually for the same species and they look very different. So I think we do get an idea from this what kind of shapes they have, which is useful to draw conclusion 1 and conclusion 2. But the exact curve for an exact species, I think that we cannot say very surely. I think this was a good way to get the overview of the possibilities, but
in the previous year, before this, you actually derive this trade-off relationship, right?
Yeah.
So, which because of which is hyperbolic, so you expect basically the kind of you don't expect any coexistence there, right? Or evolution coexistence, because it basically comes to the point where this has the maximum curvature.
Yeah, you mean you just go here? Yeah.
Well, I was more looking at this. That you have this slide where you went from,
from
the lower level to So this, yeah, the one.
I think even earlier or not.
No. The one where you went with the 4 little graphs.
Yeah.
This. No,
Ah.
that one. Yeah.
Oh, is it? Yeah.
So here you have the feasible phenotype space.
Yeah.
And this is a trade-off relationship that is that has a curvature where you would expect basically evolution to the
Yeah.
to the
It depends, of course, a bit, because they are not linear. Those are not linear properties. Huh? They are mu max and 1 over ks. So it depends. That's not.
Oh, okay. Yeah.
So it's not necessarily the case, I think. But I think, yeah, so I said it as kind of a hypothesis that you need kind of a concave space in the trade-off core.
Yeah.
I think, but the question is what exactly has to be on the axis and in what kind of. I tried even other units for these axes because I wonder what's the right unit, because it's not, of course, if it's all linear, then it becomes more easily, but in this case, I'm not. So I do think that there's some condition like that, but the exact condition, I don't know. I didn't know how to find the exact condition.
Well, but 1 over ks has exactly it's a scaling of the resource. That has ks is the same scale units as the resource density.
Mm-hmm.
So you actually could scale everything, I think, with ks for every. So that means that for a particular species, 1 over ks is actually kind of scales with the minimum resources. So that means.
Mm-hmm. Yeah. Yeah.
And in that sense, I think they are not the way you plot it here is very natural.
Yeah, and of course, this was a kind of way plot. Not these are the real plots, but still they also look a bit. Although this does have like the problem is that here the negative curvature, let's say, is at so low mu max, because mu max has much more effect than ks. That actually I did also some plot data. So from I did some other things with the selection coefficient, because you can also calculate the gradient, the selection gradient for the different parameters.
Yeah.
And for mu max, the selection is always 1. And for ks, it's between 0 and 1, depending on the parameters.
Yeah,
so
that's because you integrate it out to infinity. Because then you integrate it out to infinity, in which case you have basically converted all your substrate into resource, into biomass.
But if you integrate shorter, it's only a benefit for mu max even more, because then you don't. The last bit of the substrate is taken up by the low substrate specialist. So if you cut it off earlier, you benefit the high substrate specialist.
Yeah, you got it.
Yeah, so I don't think that matters. So yeah, but I have, I do very good. Oh, yeah, here I plotted for example the selection gradient. So the yield is always 0. I didn't discuss it at all. The mu max is always 1, and this is the selection gradient depending on the ks. And this is about the max of the reported range. This is also a reported range, and this is then for different initial glucose levels. So for very low glucose, well, you can get a decent initial, but this is like, but the normal level, maybe. So this was a thing where they once calculated this numerically in this article, and then they found it was almost 0. So in many settings, I think the selection on ks is rather low. So you need a trade-off that has a negative curvature in an area where the mu max is not too much lower than the maximum mu max, let's say. So it's not, it would be nice to have like a specific condition on the trade-off curve that says there's coexistence, but that's probably very hard.
All right, maybe it's a good time to thank the speaker again.
Yeah.
We have lunch downstairs. Please go. Thanks, everybody.